Question number 1. During the pollen grain formation, the generative cell divides to give rise to the two male gametes. What is the ploidy of the generative cell? Ploidy means the chromosome number of the generative cell. Here you can see that a microspore mother cell turns into a pollen grain. So the microspore mother cell, which is 2N, will undergo meiosis to form the microspores, which are N. So the microspores undergoes mitosis to form the mature pollen grain, where the generative cell divides into two male gametes here. So the ploidy level will be N. So the answer will be N. Kiwi is a dioecious species. Dioecious means it is unisexual. That means either the plant will have the male part or the female part. Okay, so it will have male flowers or only female flowers. So which of the following methods can be definitely ruled out as a possible mode of pollination in its case? Cleistogamous autogamy and chasmogamous autogamy. In both the cases, autogamy means it is self-pollination taking place. Okay, that means it is from it takes place in bisexual plants. Heterogamy is also a kind of cross-pollination can take place, but at the same time, on the same plant, the male and the female flowers will be present, but that is not dioecious. So we can rule out P, Q and R. Cynogamy is the transfer of pollen grains from the flower of one plant to the flower of another plant. So that is the possible way in kiwi. So the answer will be P, Q and R. Arun thinks that identifying the exact mRNA sequence from the protein sequence is difficult. Okay, so you have a protein sequence with you. From that, if you want to decipher the code of mRNA, it will be difficult. What is the reason? Because the genetic code is degenerate, because there will be more than one codon on the mRNA which can code for the same amino acid. So even if we have an amino acid and the code can be different on different mRNA, so we will not be able to know which is the right one. So that is the correct answer. Crickets are insects that follow the XO type of sex determination. XO means male heterogamety, okay, where the male chromosomes, the O will be showing on the male chromosome. Which of the following statement is always true about this type of sex determination? So it's the male part. So sperms having an O chromosome will give rise to a male cricket. Oysters are generally either dark or light in color. Dark oysters excel in dark environments, while light oysters thrive in bright environments. This type of evolution is called as the directional evolution, because it's going either to one side. Intermediate colors oysters are disadvantaged, lacking effective camouflage in either setting. When it is either setting, that means this side as well as that side, it's a disruptive kind of evolution, natural selection taking place here. A team of archaeologists found a fossilized skeleton of human-like creature with a brain capacity of more than 700 cc. The structure and its associated findings also show evidence that this creature could use tools for hunting. So which is the one, which stage of human evolution is this creature not from? So the options are these, and we know that Homo erectus, Homo habilis, Neanderthal man are the ones which are almost close to the structure of a human being, which are able to use the tools. So the option will be D. Here you can see the picture. So all of these are the ones which are almost like a human being, which can use tools, whereas the Australopithecus cannot. Which of the following is correct about the movement of DNA on an agarose gel and the reason for it? So DNA is negatively charged and therefore it will be kept at the negative terminal, that is at near the cathode, on an agarose gel, so that it will get attracted to the opposite charge, that is the positive charge, the anode. Therefore the movement always takes place from negative to positive end. And what is the reason? Because of the charge on the DNA. So the answer will be S. What is the minimum possibility of a dominant trait being expressed in the offspring after a test cross? A test cross is to find out the unknown genotype of a dominant trait. If it is a dominant trait, it will be capital T, or capital T, small t for the trait height. So when you are going to cross it with its recessive trait, that is small t, the minimum chance will be 50%. Which process is responsible for increasing the percentage of alcohol in whiskey after fermentation? It will be distillation. What does the Roman number 1 in the restriction enzyme named HIN-S2-1 indicate? The 1 is the order of discovery. So it is the first enzyme which is isolated from the strain S2 of the bacterium. Sumi and Nisha said the following about somatic hybridization. What is somatic hybridization? It is the fusion of the protoplast to create a hybrid cell which carries the traits of both the cells. Okay, and it is taken, these cells are taken from the normal tissues of the plant. So Sumi says gametes are not required for hybridization. Since we're taking normal cells, not the gametic cells, this is correct what Sumi said. Nisha said the resultant plant that grows after the fusion of the cells is genetically identical to the parent plants. We cannot say it is genetically identical as it is a hybrid plant. So who is correct? Only what Sumi said will be correct. Rupal says that in marine food chains where the pyramid of biomass is inverted, the 10% rule of energy transfer is not applicable. Is she correct, and why? We know that in the pyramid of biomass, and when you consider a marine environment or food chain, it will be an inverted pyramid. Okay, but the 10% rule of energy transfer is applicable to any food chains. So what he said is not correct. Okay, because every level still gets 10% of energy from the lower level. Assertion: the coconut endosperm is multinucleate. Okay, that is that free water that we find, as well as we see the kernel also inside a coconut. The coconut endosperm is multinucleate throughout its development because some endosperms undergo free nuclear division without the formation of distinct cell boundaries. Both the sentences are correct and they are supporting one another. So both A and R are true, and R is the correct explanation of A. DNA ligase is not used in PCR. Yes, DNA ligase we don't use in PCR. Why? Because during the formation of the opposite strand, discontinuous fragments are not formed in the application of DNA by PCR. It's correct. So the answer will be both A and R are true, and R is the correct explanation of A. To promote sustainability while minimizing waste, it is recommended to use needles up to 2 times for the same person. No, it is not correct because it affects the hygienity and transfer of diseases like AIDS, or any time such diseases can be transferred. whereas sterilization of needles eliminates all pathogens and ensures safety. But that is correct. So here the option will be A is false but R is true. Nuclear DNA extracted from a cell is visible to the naked eye, but unstained plasmid DNA running in an agarose gel is not. It is a true sentence. Reason: plasmid DNA is transparent, but nuclear DNA is not. No, that is not correct. So the option over here is A is true, but R is false. Kavya says that the placenta produces relaxin. The placenta produces relaxin, which plays a crucial role during pregnancy. Is she correct? No, it's a wrong sentence because the relaxin is not produced by placenta, but it is produced by the ovaries. Name two other hormones secreted by the placenta during pregnancy. Here you can see that the placenta can act like an endocrine gland which is able to produce certain hormones like human chorionic gonadotropin, that is HCG, human placental lactogen, that is HPL, estrogens and progestogens. But relaxin is produced at a later stage of pregnancy by the ovaries. So the hormones produced by the placenta are estrogen, progestogens, HCG, HPL, etc. Thalassemia is an autosomal recessive disorder that causes anemic conditions in an individual. A blood smear from heterozygous individual shows blood cells that are small, pale, and irregularly shaped along with the normal RBC. So, state the genotypic and phenotypic ratios of the offspring born to a carrier mother. A mother is carrier, so it will be capital A, small a, or you can take any letters for this. And a thalassemic father, the father is affected, so it's small a, small a. So the gametes, let's take the gametes out. These are the gametes. And the two offsprings that will be produced will be a carrier, which is a normal child, and an affected child. So it is 50% chance of 1:1 ratio. So the answer will be, the genotypic ratio will be 1:1 and the phenotypic ratio will also be 1:1. Does the allele for thalassemia exhibit codominance? Yes, because you see in the blood you can see both the irregularly shaped ones are there as well as normal RBCs are there because of the presence of both of them, and both are being expressed here. Therefore, it exhibits codominance. Explain any two reasons why the treatment of AIDS is only partially effective. AIDS is caused by HIV, and the mutation rate of the HIV virus is very high, and also the genetic variability making it difficult to target. HIV can be also latent, that means it will be in an inactive state for many extended period of time, making it difficult to diagnose the disease. And as it replicates in the immune cells, that is the macrophages, the Th cells of the human body, targeting them might pose a risk to the individual's immune system and safety. So these are the reasons. Rati wants to grow a variant of the Lactobacillus species in a bioreactor. Lactobacillus is an anaerobic bacteria commonly used as a starter culture for dairy products. So you can see here the structure or the diagram of a diagrammatic representation of a biofermenter, and identify one component that should definitely not be present in the reactor to grow the lactobacillus. So since it is an anaerobic bacterium, definitely the air inlet should not be there, or even the sparges should not be there. Explain two quantities that the sensors in the bioreactor should monitor. Any microorganism needs an optimum condition for its growth. For example, an optimum temperature, optimum pH. So the temperature should be controlled, the pH should be controlled, and also the amount of dissolved oxygen should be also measured, so that we should ensure it is an anaerobic condition. Shown below is a food chain, that is fallen leaf to millipedes. Millipedes will be degraded by the fungus and then the earthworm and then the soil. So millipedes have a hard exoskeleton. That means the exoskeleton is made up of chitin, whose composition is different from that of the leaves. Considering all other, and the leaf, the skeleton or the outer structure is made up, of the cell wall is made up of cellulose. Considering all other conditions to remain the same, which step is likely to be slower between A to B and B to C? Definitely digestion or disintegration of the cellulose is much simpler than that of the chitin. So for the fungus to help in that disintegration process will be difficult. Therefore, B to C will be slower, as the exoskeleton is made up of chitin, so its degradation will be difficult. What would be the direction of the flow of energy in this food chain? Flow of energy is always from A to B, B to C, C to D. A coral reef can be regarded as an ecosystem. Ecosystem is the interaction of the different kinds of species with its environment in a habitat. So, mention any two reasons why. A coral is self-regulating and a stable system. So, we know that a self-regulating means on its own it is able to maintain that habitat. So, what is the possible reason behind it? It has a variety of both biotic components and also abiotic components are present here. Okay, so what are the biotic components? The corals, fish, invertebrates, algae, bacteria, plants, animals are all present in this coral, as well as abiotic components like water, sunlight, nutrients, soil, everything is there. So both of that will interact with each other for the life to sustain. And also during this interaction, flow of energy takes place, flow of nutrients takes place. Okay, and therefore it is said to be self-sustaining system, as there will be production of food will be there through photosynthesis, and it also provides a stable habitat for the organisms to live. The net primary productivity (NPP) of a coral reef is supposed to be approximately 2000 grams, and the gross primary productivity, that is GPP, is 4000 grams. So they're asking to calculate the respiration loss of this system. The formula is GPP minus R is equal to NPP. So the GPP given is 4000 minus R is equal to NPP given is 2000. So the respiration loss will be 2000 grams per cubic meter square per year. A biologist sees the following cells in a cross-section of the seminiferous tubule and its surrounding tissues and counts the number of various kinds of cells. So let's look into the process of spermatogenesis first. A spermatogenesis is the formation of the sperms or spermatozoa from the spermatogonium. So spermatogonium primary spermatocyte are diploid in condition, whereas secondary spermatocyte spermatids spermatozoa are haploid in conditions. And in the seminiferous tubule also we find the Leydig cells, Sertoli cells, which are also diploid in nature. Okay, so here we can see spermatozoa are haploid, whereas spermatid is also haploid. Primary spermatocyte is diploid. Secondary spermatocyte is haploid. Leydig cells are diploid. Serotuli cells are diploid. And the spermatogonium is diploid. So which are the cells that are diploid? Spermatogonium, Serotoli cells, Leydig cells, and the primary spermatocyte. Now, that identify the cells that can produce hormones and their names. So amongst these, the ones that produce hormones are the Leydig cells, and they produce the male hormones, that is the androgens or testosterone. A couple is trying to conceive and start a family. If a woman's period, which is regular, is scheduled to start on July 19, what was the estimated date of ovulation for the previous cycle? So, a normal period usually takes within time 28 days time, and therefore the mid cycle, that is during ovulation, will be around 14th day. So if you count back, it will be July 5th. Count back to the 14th day, it will be July 5th. Name the four important reproductive hormones and state whether their levels will be high or low on the date identified in A. So the four hormones responsible during that time will be estrogen, progesterone, the LH and the FSH. LH, FSH and estrogen will be in very high amounts or high levels, whereas the progesterone is not present at this period of time. As part of the assisted reproductive technologies, what is the destination for blastomeres with a count of less than 8 cells? A count of less than 8 cells are usually implanted into the fallopian tube, and more than 8 cells are usually planted into the uterus. What could be the reason behind transferring to the destinations? The blastomeres more than 8 cells are much healthier compared to the less than 8 cells, and therefore can be put directly into the uterus so that it can get implanted onto the uterine wall. What techniques are used to transfer the blastomeres to the testes identified in A is the answer for that. And what are the techniques? The techniques are less than 8 cells, it's called a ZIFT, and more than 8 cells is IUT. A population of 100 individuals has a frequency of allele A of 0.3 and a frequency of allele small a of 0.7. The equation for the Hardy-Weinberg principle is p square plus 2pq plus q square. P square stands for the dominant alleles, q square stands for the recessive alleles, and these are the heterozygotes, the 2pq or the capital A small a. And here the frequency of the heterozygotes genotype is shown as 0.49, and if you applied the 2pq value with 0.3 and 0.7, you get a value of 0.42. Since the frequency is not matching with that, see this is 0.42 and in the question is given 49% or 0.49. So it deviates from the Hardy-Weinberg principle and it is not having that equilibrium. State whether each of these statements given below is true or false and justify your answer. Frogs reduce the pollution in water by increasing its BOD level. Biological or biochemical oxygen demand is the demand of oxygen by the microorganisms in a water. So if the demand is more, that means the pollution is more. The flocs are actually the collection of bacteria along with fungus which decompose the organic matter in the wastewater. So they reduce the pollution, and that means they will decrease the biological oxygen demand, and hence it's a false statement. Mycorrhiza is an association between the fungus which grows in the roots of higher plants. So they both will benefit from each other. Hence it is a symbiotic relationship, not a parasitic relationship. Erythropoietin is a glycoprotein hormone that is otherwise naturally produced in the kidney when the body becomes anemic. However, this does not happen in the case of chronic renal diseases where the kidney function is lost here. So that means they will not produce the erythropoietin. Erythropoietin alpha is a human erythropoietin produced in cell culture using recombinant DNA technology. The cell culture used is called as Namalva cells, human cell culture. There are 8 exons and 7 introns in a single gene that encodes the hormone, whose sequence is known. Explain the step-by-step process that should be followed for producing the human erythropoietin in culture. So the steps of RDT is actually what they are asking over here to produce that erythropoietin. So the first thing is you are going to use that exon, the sequence of exons to form the double-stranded DNA. This is inserted into an appropriate vector, and the vector is introduced into the bacterial host cells, and you're going to select it, the recombinants from the non-recombinants, okay, and purify the recombinants or the recombinant vector, and you can amplify it using PCR technique, and then you put it into the human host cell, which is the Namalva cells. And if you want to produce the erythropoietin in large amounts, use the biofermenters, bioreactors, and make these cells to produce the hormone in large amounts, purify it from the culture medium. Today, many genetic disorders can be detected using a single cell from an embryo. Okay, it is about the genetic disorder detection. This helps in planning the child's health care in advance, and in some cases even treating the disorder while the baby is still in the womb. Identify a biotechnological technique that can be used for this purpose and give a reason to support your answer. ELISA and PCR are techniques that can be used for early detection of disorders. And here we can use PCR to amplify the DNA sequence of such children and early detection of that disease. Can this technique be identified in A, be used to detect the presence of RNA viruses and justify? Yes, it could be, because both DNA and RNA are nucleotide sequences, and PCR is a technique which can amplify any nucleotide sequences. In a study comparing two continents, Antarctica and Asia, the species area relationship was given as given in the table. So they're asking you to find the species richness value and which continent will have a greater biodiversity. So the equation for finding species richness, learn it by heart. S is equal to Ca raised to Z. S is the species richness, C is the y-intercept, A is the area, and Z is the regression coefficient. So just apply the values here for Antarctica and for Asia from the table, and you will get the answer here. So which continent is having a species richness? So here you can see Asia has a more value, that is 440 into 10 raised to 6 or 44 into 10 raised to 7. Whereas here it is only 70 into 10 raised to 6. So more the S value, more will be the species diversity. So Asia will be having more, since the species richness of Asia is more, as they have more species and so greater biodiversity than Antarctica. Here is a picture of a vector, a recombinant vector with the gene of interest. Okay, and there are certain sites where you can see there are two origin of replication here. There are sites for EcoR1. This is a selectable marker with chloramphenicol, and here also there are restriction sites for HIM3 over here, SCAR1 here. So this is a recombinant vector. The vector consists of sites for 3 restriction enzymes, SCAR1, HIM3, and EcoR1. Restriction sites for the same enzymes are also present in the gene of interest. There are 2 origin of sequences. One it allows to replicate in the E. coli and the other one in Cymomonas nobilis. That is, one in a bacteria and one in a plant. Apart from this, the vector consists of 2 antibiotic resistant genes. One is tetracycline resistance and the other one is chloramphenicol. What is the advantage of having 2 origin of sequences? 2 origin of sequences help in replicating that in both bacterial as well as plant cells. Give a situation in which this would be particularly useful. So when you try to put in a bacterial genes and you're going to insert it into plant vectors, Having the origin of replication for both, we can make them to replicate both in bacteria as well as in plants, and this will simplify the process of gene cloning. If the vector was cut using Hin3, what would colonies growing on a medium containing tetracycline definitely contain? So if you're going to cut this vector with Hin3, and grow that vector containing cells in a medium containing tetracycline, definitely the recombinants will die and only the non-recombinants will be present, because the tetracycline will be made inactive as the Hin3 is present over here. This part will be removed and hence no tetracycline resistant gene, and hence it will not grow in a tetracycline medium. Suggest which of the three restriction enzymes would be suitable for insertion of the gene of interest, and give two advantages of using the enzyme chosen by you. So there are three restriction enzymes, CAR1, HIN3 and EcoR1. So I would prefer to choose EcoR1 since there is only one site where it can cut compared to Hin3 it has two sites, CAR1 has two sites. Therefore multiple pieces will be there and it leads to confusion. Hence EcoR1 having only one site, it will not produce multiple products and you're able to insert the gene of interest. So you can get only either the vectors with the gene of interest or without it. Second reason is, since the restriction site for EcoR1 is within the chloramphenicol resistant gene, it helps in selecting the recombinants from non-recombinants by insertional inactivation. Suggest one disadvantage of using the other two restriction enzymes not chosen in B. If you use Hin3 or SCAR1, it can produce multiple fragments and therefore selection becomes very difficult. Predator Y. This is the predator population shown and this is the prey population shown, which is denoted by X and the predator by Y. And the predators are the wildcat which eats the herbivores shown here. What is the likely cause for the pattern seen in the prey and the predator population over the years? So through the years what we find is that when the prey population, the predators feed on the prey and prey is the primary source of food for the predators. Therefore, when the predator population when the prey population increases, the predator population also seems to increase. And when the predators consume the prey population more than the number of prey, the prey population begin to decrease. And when the prey population begin to decrease, the predator population also decreases as they don't have any food. And when the predator population decreases, now the prey population can increase. So that is what happens over time. It continues. Hypothetically, if all the predators of the forest become extinct, what will happen to the vegetation of the forest? Definitely, if the predator populations is not there, then the prey population will keep on increasing, eating on the herbs or the grass, and therefore the vegetation will decrease. Consider a situation where another similar species of predator immigrates to the forest. What is likely to happen over time and why? There will be a competition between the predators for the same species, and over time the inferior one will, out of those predators, will go extinct and the other one will survive as the resources are limited here. Juglone is a chemical produced naturally in most parts of the black walnut plant. This chemical leaches into the soil when the plant falls. This leads to the death of many plants that grow around the black walnut plant. Identify the type of ecological interaction. It is called as amensalism because the black walnut, by producing the chemical, neither benefits or has any nor any problem happens to that plant because of that chemical. But it is at the same time destroying the plants around it. So this kind of interaction is called as amensalism. A non-government organization aims to increase awareness against STDs, that is sexually transmitted diseases. What could be the ideal target age group for the NGO? 15 to 24 years of age. Mention any two potential long health related complications of untreated STDs that the NGO should educate the target age group about. People suffering from long-term STDs can have discomfort in various body organs, infertility, chances of transmitting the diseases, lower immunity levels, catching diseases very fast, body pain. These are some of the symptoms. Mention one contraceptive method that provides protection against the STD. So condoms justify. The condoms, they act as a barrier preventing the mixing of the body fluid. State two contraceptive methods that do not protect against STDs that they can educate the group about. It is oral contraceptive pills or IUDs or vasectomy, etc. Amy and Lalita are expecting their first child, and Lalita being in her second month of pregnancy with no known complications. Amy's family has a history of cystic fibrosis, while Lalita's family has a history of Down syndrome, leading to a concern that the baby may have one of these conditions. Suggest and explain a way of testing if their baby is at risk. So the method for testing in such case when the baby is inside the womb itself is amniocentesis. In case of the presence of one or both of the abnormalities and posing a risk to the mother's health, mention one possible option for them to consider, that is MTP, termination of the pregnancy. Is the process mentioned in B safe for Lalita at the current gestational age before the first trimester? It is always safe, so it is okay. Under what conditions is the process mentioned in B illegal? MTP is illegal in cases involving determining the gender of the unborn child and female feticide. Okay, in order to know the sex of the child, and if it is a female, killing of the child, it is illegal. Shown below is a pedigree of an individual X who is suffering from ocular albinism, which results in permanent vision loss. Use the pedigree to answer the questions that follow. So all these green and red ones are not given there. That is the answer which I have written here. Okay, so only the picture is given and with the RPQ as an X. Okay, is the question. So first of all, you have to look into two questions, whether it is a sex-linked disease or is it an autosomal disease. If it is autosomal, both males and females will be affected in every generation randomly. So here we can see that it's not a randomly. Most of the males of the generation are affected here, and female by chance only one is there. So that means this is a sex-linked disease, and sex-linked diseases you have to write it on the X and Y chromosomes. Otherwise you can write the alphabets directly. And second question that you have to look into is whether it is an autosomal or a recessive disease. Okay, an autosomal disease means every dominant or recessive disease. Dominant means every generation will have it. They will not skip any generations. So if you look into this, the first generation is having, and the second generation, most of the children are not having, one child is having. Third generation, one child is having. And when you look over here, you can see that these parents are having a child with an affected case, but the parents are not having it. That means it skipped a generation here. Therefore, this is a recessive disease. So the trait for the disease is linked to what kind of disease? It is an X chromosome disease because only the males are majorly affected and the X chromosomes are affected here. Okay, so with the XY is the sex chromosomes for a boy, and that A which is written as the affected allele. Over here also you can see, and the case here also the X is affected, only one of the females, but majority of the children are not affected. So it is a sex-linked disease. And the second case, it is a recessive disease, since you can see that a child is born where the parents are not affected here. So the reason I have said right now, identify the genotypes of the individuals P, Q, R and S marked in the pedigree. So the first thing that you have to write is the sex chromosomes. You write on all the individuals given here, XY, XX, like that. And the affected individual, here it is the small a, which, since it is a recessive disease, you have to put small a. Okay, and only one X chromosome is required for that particular males to get affected. Here it should be the both of them should be affected, and if this child has to be affected, one of the X chromosome here will be of small a. One of them will be small a and the other one will be capital A. So here, and therefore both the X chromosomes should be affected for a female to get this disease. All the rest are given here. This is how you will write it, and that will be the genotypes that you have to write. Shown below is a nucleotide sequence and the genetic code is given over here. So the nucleotide sequence is given from 3' to 5', and if you need a genetic code, it will be present on the mRNA. So transcription of this DNA to mRNA should take place, which is from 5' to 3'. Always a new mRNA strand will be formed from 5' to 3'. Just write the complementary with, instead of T you have to write U. That means if A is there, instead of T you will be putting it as U. So complementary to A is U, T is A, and so on. You will write the sequence. So we have the mRNA sequence ready, and a set of codons are present here. A codon is a set of 3 nucleotide bases which codes for one of the amino acids. Here is a list of 20 amino acids and the code is given here. So identify the protein sequence formed by this sequence is what they asked. So here is your mRNA. From this, what kind of protein will be formed? So each 3 set will make 1 amino acids. Like that, so many amino acids. All amino acids together make a protein. So let's see. The first one is, the code is UAC. So U is here, A is here, and the C is here. So it will be tyrosine. See, UAC. So what is the code over here? The amino acid tyrosine. So the first amino acid is tyrosine. Next one is GCA. So G is here, C is here, A is here. So which is it? Alanine. So the next one is alanine. Next is UCU. U, C and U is here. So which is it? Serine. So you can see serine. So likewise you have to find for the rest. Glutamic acid also I'll show you. GAG, G, A and G is here. Okay. So it is once again G, A and G over here. Okay. So wherever it's starting, that particular column you have to look into, and the central base region. GAG is, CA is coming here, so it is glutamic acid. Histidine is CAU. So let's see, CA and U is here. So here, histidine. So that's how you find the amino acid sequence. So this is the protein sequence. Draw the tRNA molecule for the third codon. The third codon is UCU. So the anticodon for the tRNA will be having the anticodon arm, which will be complementary to the mRNA. So the third codon UCU will be, the complementary base will be U versus will be A, C versus G, U versus A, AGA. And the polarity will be, since this is from 5' to 3', the next one will be 3' to 5'. So 3', AGA, 5'. This is the tRNA sequence with the anticodon region and the polarity. The first guanine base in the nucleotide sequence changes to cytosine. That means if you look into this DNA nucleotide, they are saying that the first guanine has changed into C. That means ATC. If this is changed, then the codon over here will change to what? UAG. Instead of C, it will be G. So if it becomes UAG, identify the type of mutation caused by this change. That one single base is changed here. Such kind of mutations are called as point mutation. Will the mutated sequence form an mRNA? Yes, it will form an mRNA. It will become UAG and the remaining as given here. And will the mutated sequence form a protein? No, it will not form a protein, since the first amino acid will not be formed here because it's a stop codon. UAG is a stop codon. UGA, UAA and UAG are stop codons. So at the starting itself it stops, and therefore no protein chain will be formed here. That is, no translation will take place to form a protein. Classify the following scenarios as active or passive immunity and justify your answer. A fetus receives antibodies from its mother through the placenta. It is called as passive immunity because ready-made antibodies from the mother through the placenta is being transferred. The child is not making its own antibodies there. A person accidentally gets cut by a blade and later receives a tetanus shot. A tetanus shot is a shot which contains the tetanus toxin, which is injected into that person's body, and the person is going to make the antibodies against it. So it is active immunity. So inactivated tetanus toxin will stimulate the person's immune system to produce his own antibodies here. So it is active immunity. A person receives a blood transfusion from a donor who has been vaccinated against a disease. That is passive immunity, since his body is not making that antibodies and he is receiving the blood which already has those antibodies. So it is passive immunity. Soya is bitten by an infected Anopheles mosquito in the morning. In the evening, another non-infected Anopheles mosquito bites Soya and then bites Sahir immediately. How likely is Sahir to get malaria? And justify your answer. So if an Anopheles mosquito bites, through the saliva into soya, the sporozoite forms or a sporozoite form will be going into their body. But it takes some days for the infection to progress, and therefore immediately he will not catch the malarial disease. Since soya has been bitten by an infected mosquito, the process of infection of the liver cells and the red blood cells takes more than 5 days, the non-infected second mosquito is not likely to get infected by biting soya and thus cannot transfer the plasmodium to Sahir. The patient is suffering from fatigue, high fever, weight loss, and an increasing number and size of lumps, you can say. This can result in cancer. What could she be suffering from? It is a cancerous disease or malignant tumor. Mention 4 ways in which the disease identified in A is caused. So what are the reasons for getting this kind of cancer, and 4 techniques that can be used to identify it. So it can be genetic factors or family history, or the use of carcinogens, high energy ionizing radiations like X-rays, gamma rays, non-ionizing radiations like UV rays, or oncogenic viral infections can also cause cancer. The various techniques to find it is X-rays, CT scans, MRI scans, biopsies, blood tests are some of the methods to identify the disease.